Okay, so let's get started. Hello, everyone. Welcome to our today's Museum seminar. It's really my great pleasure to welcome our today's speaker, Dr. Eui-Cheol Shin from Korea. So Eui-Cheol currently is full professor at the Graduate School of Medical Science and Engineer, which is belong to Korean Advanced Institute of Science and Technology, which is short as a KAIST in Korea. I believe this is one of the best funded and then best advanced institution in Korea. So Ichun actually completed his MD training in Yonsei University College of Medicine, and then after which he pursued his PhD training in microbiology and immunology, also in Yonsei University College of Medicine. So after complete his PhD training, he actually moved to U.S. Joined NIH as a research postdoc fellow in immunology section in liver disease branch with NIDDK, then supervised by Dr. Barbara Hannes, and then from 2007. Ichun actually moved back to Korea, started his independent research program in as assistant professor, all the way up to full professor in the laboratory of immunology in the infection disease in KAIST. As I mentioned, that currently he's a full professor in the KAIST, but at the same time he also have exert as a director of Center for Viral Immunology in Korea Virus Research Institute. So the main interest of Yichun's lab is viral immunology, tumor immunology, immune agent, and human immunology. Um, his lab investigates T cell responses in human viral disease and cancer with a focus on cytotoxic T cells, and then particular interested in T cell mediated immunopathogenesis and also senescence of T cells, invariation of exhausted T cell and immune responses in the SARS-CoV infection. So, particularly during the COVID pandemic, his research has focused on functional characterize SARS-CoV-2 specific T cell following infections, vaccination, and breakthrough infection. Um, so, actually, because of all these achievements, Ishan actually being elected as a fellow of Korean Academy of Science and Technology. Also, he's a fellow of the National Academy of Medicine in Korea. So it's really our pleasure to have you on our platform, Yuchen, and then we are very much looking forward to a talk.
Yeah, thanks very much for your kind introduction, and thanks for inviting me to the Immunozoom series. And today, I'm going to talk about the NK-like activation of CD8 T cell. In other terms, I can say that bystander T cell activation. During the viral infection, as you know very well, the virus antigen-specific T cell will be activated by TCR stimulation. But in certain circumstances, not only this virus antigen-specific T cell, but also any other pre-existing memory CD8 T cell can be activated without TCR stimulation, without antigen, but by cytokine. We call this phenomenon bystander T cell activation. 7 years ago, so in 2018, so in my lab, so we published one paper about the medical significance of this bystander activation. This bystander T cell activation, it was known since 20 years ago, not 20 years ago, yeah, around 20 years ago. But meanwhile, the medical significance was obscure, not so clear. But 7 years ago, by analyzing this patient with the hepatitis A virus infection, HIV infection, we found that what's the meaning of this bystander T cell activation for the first time. Let me summarize very briefly this the previous publication. During HIV infection, IL-15 will be overproduced. Then this IL-15 can activate non-HAV specific, any kind of pre-existing memory CD8 T cell. Then this the T cell independently activate CD8 T cell. They upregulate NKG2D or NK-30. Then in the liver, this the bystander activate CD8 T cell, they can kill this the target cell, hepatocyte, even without any antigen, without cognate antigen by NKG2D or NK-30. So we can say that NK-like or innate-like cytotoxicity of bystander CD8 T cell. So in this way, in that time, we show that these the bystander activate CD8 T cell, they can contribute, kill hepatocyte, they can contribute to the immunopathology, the viral hepatitis, inflammation itself. So let me show you some more slides from the previous publication. So this one is the definition of bystander T cell activation. In this case, HIV-infected patient, we apply the HIV tetramer, and then we analyze the activation status of those T cells, CD38, HLA-DR. All of them are very highly activated. Well, this is expected data. But not only this HIV specific, but also the CMV specific, the CMV tetramer. CMV specific tetramer, also they are highly activated. These are from the liver sample, liver tissue, and not only liver tissue, but also we found in the blood sample, this one is the y-axis is the activation status. The HIV tetramer positive cells are highly activated, but not only HIV, but CMV, EBV, the influenza A virus tetramer positive cells are highly activated in HIV infected patient compared to healthy donor. These are the definition of bystander activation. And then we also, not only just the activation marker, we also perform the NK-like cytotoxicity. In this case, we sorted CD8 T cell from the infected patient, infected liver tissue, and then we co-culture this the CD8 T cell with K562. K562 cells are typically the NK target cell because they don't express HLA class 1. So they cannot be target of T cell. But we found that the CD8 T cell sorted from the infected liver, they can kill this K562. So it means that they can kill the target cell without HLA class 1, without TCR. So we found that NKG to the NKp30 upregulated in this cell, and we perform again this NK-like killing activity in the presence of the blocking antibody. And we found that NKG2D, NKp30 is really responsible for this the NK-like killing activity of CD8 T cell. So in this way, we could say that during the certain viral infection, the CD8 T cell can be activated by, without TCR, then they can kill target cell like NK cell. And then we found that this the NK killing activity nicely correlate with the serum ALT level. It means that this the bystander activation, it is associated with this the tissue injury during the viral hepatitis. Finally, in this our previous publication, we found that IL-15 is upregulated, and then we could recapitulate this phenomenon by IL-15 treatment, in vitro treatment. It means that in this figure, not this CD8 T cell, effector cell is not sorted from the patient sample. We sorted memory CD8 T cell from healthy donor and stimulate them with the IL-15 in vitro. Then we perform this the killing assay against K562. And then this the NK-like killing activity could be abrogated by NKG2D or NKp30 blocking antibody. It means that, so like this, the summary slide. So in any kind of viral infection, in theory, in principle, the pre-existing memory CD8 T cell can be activated by overproduced IL-15. Then this IL-15 treated the CD8 T cell can upregulate the NKG to the NKp30, and they can kill target cell without any antigen by NKG to the, like NK cell. They behave like NK cell. So after this publication, my lab, we continued this the bystander research. Then once we also found that IL-15 upregulates not only NKG2D, but also CCR5. Also, we showed that by the action of the CCR5, the bystander activated T cell, they can migrate to the liver environment in the patient with the viral hepatitis. And also we found the negative molecular regulator of this the signal. We found that CD5 is the negative inhibitor regulator of this bystander activation. And also we found that why, how this IL-15 is overproduced during viral infection. Before our this publication, many people believe that only myeloid cell produce IL-15 and they can transpresent IL-15, but not only by myeloid cell, but also we show that epithelial cell, like hepatocyte, can also produce IL-15 and IL-15 receptor alpha by the stimulation with type 1 interferon or type 2 interferon. So, and then also action mechanism is the IRF1. So in this way, we also study this kind of mechanism. And then this our first publication now, more and more people research the same mechanism in other disease. First, one of the maybe yeah in Taiwanese, the Changgung University, they recapitulated our same the study, but not in HIV patient, but in HCV patient. And they found that exactly same mechanism it is working in patient with chronic Hep C. And also by collaborating with my colleague in my same department with the Dr. Su-Hyung Park, my lab also showed that the same mechanism is working not in viral disease, but in the mouse model of alopecia areata. In this alopecia areata, so the responding cell is the not true memory cell or virtual memory cell, but it's a similar mechanism, IL-15. And then in this case, the target cell is not hepatocyte, it's a hair follicle cell. So that's the reason why this patient suffer from the alopecia areata. We also show that similar mechanism. And also after this publication, human HPV infection or mouse Zika virus infection, many people showed that same mechanism is working in the various disease, not only viral disease, but also other T-cell mediated some inflammatory disease. But so now, so far, I showed you the importance of the study of bystander T-cell activation in human disease. But meanwhile, my talented postdoctoral fellow in my lab, Dr. Ho Young Lee, he found very strangest, very surprising phenomenon. So already I told you that so IL-15 can upregulate NKG2D. So this one is showing those results. For the data, we sorted the CD8 T cell from healthy donor, healthy human, memory CD8 T cell, and we stimulate them in vitro with IL-15. And IL-15, as I told you before, really upregulates the NKG2D. But TCR stimulation mimicked by anti-CD3 antibody, this anti-CD3 antibody does not induce NKG2D. But surprising part was this one. If we combine both IL-15 and TCR stimulation, then there's no NKG2D upregulation. The induction is abrogated. But this one is surprising, strangers. So why it happened? So Ho Young asked this question, and then Ho Young decided to study this responsible mechanism behind underlying mechanism. What happened? What's the interaction between IL-15 stimulation and TCR stimulation? But meanwhile, also he set up exactly the same kind of experiment, and in this case, he examined not NKG2D, but activation marker and proliferation marker. But in this case, IL-15 induced activation and the proliferation, and TCR stimulation also induced activation and proliferation. And by combination, there's some additive or synergistic effect. It means that in general, IL-15 and T-cell stimulation is synergistic or additive in memory CD8 T cell. But only in certain aspect, NKG2D, this one is counteraction. So something strange. So he concluded the first time TCR stimulation counteract certain aspects of IL-15 induced activation of memory CD8 T cell, like NKG2D. And then first, he hypothesized that maybe not only NKG2D, there's some more such kind of gene. So he won't know the landscape of such landscape. So in this case, he sorted human memory CD8 T cell again from several donors and stimulate them again, IL-15, CD3 antibody, and combination, and perform bulk RNA-seq analysis. So as you can see here, really, like cluster 7, many genes induced by IL-15, CD3, and combination synergistic or additive effect. But what we were looking for was cluster 4. This cluster 4 is induced by IL-15, not by CD3 antibody, not by combination. So now you can find such kind of gene here, induced by IL-15 and not by combination. This one is like cluster 4. There are several genes. Then what kind of genes are there in this gene set? So in the GOBP analysis, we found that in this gene set, so mainly interferon responsive gene and NK, the like killing activity gene, NK cytotoxic gene, like NKG2D. So this is reasonable. Then also in the gene set, the GSEA, we confirm again NK cytotoxicity, interferon, the responsive gene are really highly enriched in this the bystander specific gene set. And then, but still, Ho-Young, he could not explain why. So yeah, not only NKG2D, this phenomenon could be expanded to other gene, but why certain gene regulated in such manner? And then, simply, he Googled this the signal pathway, the TCR signal and IL-15 signal pathway. So you know very well this signal pathway, but he just immediately found that the many things are overlapping. But non-overlapping one, this one, calcium, calcineurin, and PAD pathway are present in TCR signal, but not in IL-15. So for the TCR-mediated counteraction, he hypothesized that maybe this calcium calcineurin the signal pathway, it is responsible for this the counteraction. So he repeated again with this the hypothesis, the same exact experiment with the sorted human memory CD8 T cell, but in this case, instead of CD3 antibody, he used ionomycin. You know very well, ionomycin is calcium ionophore. It mimics calcium calcineurin part in TCR signal. And as you can see here, really, certain gene set induced by IL-15 and not by ionomycin, but not by combination. So we could find again such kind of gene, again, and then in the GOBP exactly same kind of gene, interferon responsive gene and NK cytotoxic gene. So in this way, he could know that really calcium signal among this the TCR signal, the calcium part is involved in the counteraction of this our IL-15 induced NK-like activation. So he confirmed again the finding by flow cytometry. Really, in the human memory CD8 T cell, IL-15 stimulation, NKG to the upregulation. Ionomycin, no NKG to the upregulation. By combination, no induction. So he confirmed again. But then, what's the medical meaning of this one? So also, the Ho-Young asked this question, what's the clinical meaning? There are two meanings. The first one, simple one, is this one. So everolimus is a well-known mTOR inhibitor, and tacrolimus is a well-known calcineurin inhibitor. Everolimus and tacrolimus is a clinically widely used as an immunosuppressant. And as you can see here, very simple experiment. In the human memory CD8 T cell, IL-15-induced NKG2 regulation, it is suppressed by everolimus, but not by tacrolimus. This NK-like cytotoxicity not inhibited by tacrolimus. Sure, this data is not so surprising, expected, because IL-15, it doesn't have any calcineurin pathway. But what's the clinical meaning? In certain immunopathology, T cell are activated not by TCR, by IL-15. We don't need to use tacrolimus. So it means that so many clinicians, they use immunosuppressant in the T cell is very highly activated in this disease. They use whatever immunosuppressant. But in the case of IL-15 induced bystander activation, calcineurin inhibitor is useless, meaningless. And then second clinical significance is much more important, more complicated. So in this simple experiment, in the human sorted memory CD8 T cell, so we treat them with the IL-15 and CD3 antibody by combination. But there's no induction of NKG2D because TCR stimulation counteract this IL-15-induced NKG2D upregulation. But already I told you that the calcium-calcium pathway is responsible for this counteraction. So if we combine tacrolimus all together, then tacrolimus suppress this calcineurin pathway. So paradoxically, tacrolimus upregulated NKG2D. It means that in certain circumstances, immunosuppressant, well-known immunosuppressant, can activate, can upregulate NK-like cytotoxicity of bystander activated CD8 T cell. This is very important one. So far, this is just the short summary so far what I told you. In the IL-15 alone, in the human memory CD8 T cell, it can activate the T cell NKG2D and NK-like activation by NKG2D upregulation and other genes. But if we combine this the treatment with the TCR stimulation, this calcium calcinin pathway the suppress this IL-15-induced activation. So NKG2D would not be upregulated and they would not exert NK-like cytotoxicity. But if we combine all together the molecule IL-15, TCR, and calcinin inhibitor, by suppressing this inhibitory pathway, the suppressing the pathway, paradoxically, the T cell will upregulate NKG2D and NK-like cytotoxicity would be upregulated. So this one is a kind of clinical meaning what I found you that I showed you so far. But so far we could not identify the responsible. I did not show you in detail the molecular mechanism why this calcineurin pathway down-modulate the IL-15 induced NKG2D regulation or other NK-like activation. So Ho-Young want answer to this question. And then first of all, he want to know which transcription factor is most important for the IL-15 induced NKG2D upregulation. In the literature, he found that one of the most important one is AP1 in the IL-15 induced NKG2D upregulation. So he confirmed previous publication. So really, this is the chemical AP1 inhibitor or JNK inhibitor. So it abrogated IL-15 induced NKG2D upregulation. It means that this IL-15 upregulation is mediated by AP1. And to make sure, he once again confirmed this data by doing the June and 4th siRNA knocking down experiment. In this case, really by knocking down June or 4th or by combination, the IL-15 induced activation of the NKT upregulation was abrogated. Then with this background information, he hypothesized that with this wonderful hypothesis. So this one is only IL-15 stimulation to memory CD8 T cell, then IL-15 will activate AP1. Then the AP1 will upregulate the NKG2 then other NK-like cytotoxic gene. Then NK-like cytotoxicity would be upregulated. But then upon TCR combination, TCR stimulation will activate calcium-calcinin pathway. Then finally, NFAC1 would be activated. But it is previously well known that NFAC1 can bind to AP1. Then NFAC1 can do their full transcription factor activity by binding to AP1. So NFAC1 do something by binding to AP1. But meanwhile, Ho-Young has hypothesized that AP1 will be sequestered from the original site to this site. So it means that the AP1 alone, the NKG to this action is required. Without NFAC1, only AP1 alone. But so the AP1 binding site would lose the AP1 binding. So that's maybe reason why the concurrent T cell stimulation can down-modulate this NKG to the upregulation. So experimentally, he confirmed his hypothesis. So fortunately, around more than 20 years ago, it was well known that the mutant cannot bind to AP1. It means that this one is the wild type AP, the NFAC1. The wild type NFAC1 can bind the AP1 very well. But NFAC1 T551A, this the mutant, it can bind DNA still, but cannot bind anymore AP1. So the Ho-Young used the two different constructs for the transfection analysis. So in this transfection assay, so all the data was obtained under the IL-15 treatment. Really, IL-15 treatment and with the empty plasmid transfection NKG2D was upregulated. And then by the wild type and FAST1 transfection, this one is somewhat abrogated. And by this the mutant, the transfection, not completely, but the partially it was recovered, the NKT level. So in this way, he showed that really this the apion binding activity is very important for this the NKG2D upregulation. The TCR mediated the abrogation. And then he also showed that so by using very simple model, PMA inomycin, you know very well PMA inomycin as the mimicking stimulation for TCR. And overall, we can say that PMA is responsible for AP1 or NF-kappa B through PKC. And ionomycin is the calcium ionophore calcium and fat pathway. So in this case, after sorting human memory CD8 T cell, Hoyoung treated them without IL-15 and only PMA or ionomycin. So here's the hypothesis that in this case, in terms of NK-like activation, PMA can replace the role of IL-15. It was the case. The PMA without N-phosphon activation, the PMA alone, it could induce NKG2D upregulation. Ionomycin, it does not upregulate the NKG2D, but by combination, PMA ionomycin, NKG2D is not upregulated. Really, the fantastic. So this one, but many people before didn't know about this, the simple knowledge, because many people used PMA and ionomycin in combination to fully mimic TCR signal. So they could not know the NKG2D regulation by the apion alone without N-phosphon. So in this way, he showed that his hypothesis is working. So really, the TCR, the down modulate, the counteract this the IL-15 induced AP1, the induced the NKG2D upregulation. So in this way, concurrent T cell stimulation interfere with NK-like activation of human memory CD8 T cell. Then, so let me a little bit go back to my previous slide. You may remember this my previous slide. In my previous slide, we sorted the memory CD8 T cell and stimulate them with the IL-15, CD3 antibody, and combination. And you may remember this cluster 4, this one, induced by IL-15, but not by combination. And also, at the same time, and then when we got this data, Hoyoung, he hypothesized that maybe we can use this gene set as a bystander activation specific gene set, because recently there are many single cell data in some inflammatory disease. And then if we find that a certain CD8 T cell has upregulation of such kind of gene set, it means that there's no concurrent T cell stimulation, then only IL-15. So Hoyoung suggests that we can use this gene set as a bystander activation specific gene set. So in any other kind of the single cell RNA analysis. But first of all, we needed to validate this one in our own hands. So Hoyoung did that. So far, one of the prototype disease in which the bystander activation play a role, it is the HIV infection. What I showed you, we published 7 years ago. So we went back to this HIV infected patient sample. And then we sorted the HIV infected in acute stage, the CD8 T cell, and we stained them with decode dextramer staining, the tetramer-like one, but DNA barcoded tetramer. In this case, we use HIV tetramer, CMV, EBV, influenza virus tetramer. So then we performed the single cell RNA analysis, the more precisely site-seq analysis. So in this case, now I'm showing you UMAP. So now I'm showing you the red color is the HIV tetramer positive cell. So they are TCR, the induced activated CD8 T cell. But in this case, CMV, EBV, the flu spec T cell, they are, in this situation, they can be defined as a bystander T cell. Then really, our gene set, the bystander activation specific gene set, are really selectively upregulated in by these three population, HAV and the bystander CD8 T cell population compared to HAV specific T cell. So we also confirmed that in GSEA. So it means that by using this the specific gene set, we can easily identify is the T cell who experience in vivo situation, who experience IL-15 induced bystander activation without concurrent T cell stimulation. And 3 years ago, we also published one paper in JI. In this case, we set up the mouse model of bystander activation, so with the precise model. In this case, we set up OT1 memory mouse, and then we stimulate them, they infect them with PLA. But in this case, also in certain mice, we injected synfectal cognate antigen. In this case, so for control situation, bystander activation, only PLA flu infection without cognate the peptide, and the without infection, only synfectal stimulation, TCR activation, or combination. Very similar to our previous the data. And then, but this one is in vivo treatment, and we found that really this what we found the bystander activation specific gene set are really selectively enriched in the bystander activation situation. So even in mouse model, we can use this gene set. So this one is now I'm showing you the summary so far what I showed you. So in the IL without TCR stimulation, only IL-15 stimulation, NKG2D is unregulated, and NK-like killing activity is unregulated. And in meanwhile, we could define this IL-15-induced bystander activation specific gene set signature. You may use this one. And also, concurrent T-cell stimulation cannot inhibit the NK-like killing activity by recruiting AP1 to NFAC1. And then, calcineurin inhibitor can abrogate the T-cells contact. So now the data is open in bioarchive, and now in the revision in immunity, now this is the whole story. So I would not repeat this summary, but I have some more slide, a little bit more slide. I would not repeat this summary, yeah. Then, so this is the ongoing the project, and this one is the sure about bystander activation. But we found that this bystander activation has a close some association with the immunosenescence. So from now, I'm going to show you how immunosenescence is important in this bystander T cell activation. So this one, now I'm showing you this naive cell, one individual subject the T cell compartment, naive cell and memory cell. But in the case of human, in memory cell, there's well-known marker for the senescent memory T cell. So now I'm showing you the CD28 positive and 57 negative regarded as a young memory cell, but 28 negative, 57 positive cell, the replicative senescence memory cells, the T cell. But I told you that the IL-15 induced activation, bystander activation, so far, we use this memory cell population altogether. So now I'm showing you this the one of the representative facts plot. So really 28 positive are 57 negative, 28 negative are 57 positive. But this cell population is not discovered by our own group. It's already well known. This the 28 negative 57 positive cell, upon aging, the proportion of the senescent cell increase. And we'll skip this one. Yeah, then this one. So in our own group, we confirm again. So in several Korean, their age to 20 to 70 or 80, we confirm again that in the aged people have more higher frequency of CD57 positive memory CD8 T cell in their peripheral blood. We confirm again their senescence feature. Really compared to 57 negative cell, young memory cell, the senescent memory T cell has shorter telomere length. And also, in the senescence study, it's well known that beta-galactosidase activity is increased in the senescent cell. It was the case in the 57 positive cell. And then, in this, our some preliminary study, we sorted 57 negative young memory cell and 57 positive old memory cell, senescence memory cell. And then we stimulate them with IL-15. And then we found that IL-15 induced upregulation is so much exaggerated in this the senescent CD8 T cell compared to young memory T cell. Then how about NK-like cytotoxic activity? So in this case, again, we sorted young memory and senescent memory and stimulate them with IL-15 and perform K562 NK-like killing activity. As you can see here, the young memory cell, the blue color, their NK-like killing activity is the ignorable. But only the senescent CD8 T cell. So when they were exposed to IL-15, they showed NK-like killing activity. So the senescent CD8 T cell are much more hyper-responsive to IL-15 compared to young memory CD8 Ts. So it means that our previous publication or our ongoing previous the study, so far we showed you the bystander activation of memory, total memory CD8 T cell. But all of this, the activity might be attributed to the senescent CD8 T cell. And then this one is very interesting data. So we obtained again the CD8 T cell from the several donor, from the many donor from the age 20 to 70. And then we stimulate them with IL-15. Then, as you can expect, who will respond better? Of course, the elderly people, they have a higher proportion of the senescent CD8 T cells. So as a consequence, IL-15 induced proliferation, IL-15 induced NKG to the upregulation, IL-15 induced cytotoxic activity, everything is much higher in the old individual. And also, this one is, regardless of this age, this the IL-15 induced proliferation, NKG to the upregulation, and NK-like killing activity, all of these the parameters are highly associated with the proportion of the senescent CD8 T cell in a single individual. So with this data, now we cautiously suggest that maybe, so this one is the final point, the senescent CD8 T cell exhibit higher response to IL-15 than young memory T cell, which might explain a high degree of bystander activation in the elderly. So now we are expanding this current data to explain why certain inflammatory disease is much more severe in older individual. And at this point, I also want to emphasize that our whole the story, it began from the HIV infection, hepatitis A virus infection. Typically, clinically, you may know that HIV infection is asymptomatic and subclinical in infant or children. Only in the old individual, they suffer from the very severe liver damage. So this the phenomenon, clinical phenomenon, well fit to this our hypothesis. So this one is my final slide. So far, I showed you the meaning and mechanism of the IL-15 induced bystander activation. After my labs, the 7 years ago publication, more and more groups, they are now studying cytokine-induced bystander activation in human disease or mouse model. And 3 years ago in our review paper, we reviewed that, but there are two different types of the bystander activation. The one is induced by IL-15, what I showed you today. It induced finally NK-like killing activity. But also there are the different form of bystander activation. IL-12 and IL-18, if these cytokines are too much overproduced, then they can activate CD8 T cell without TCR. Then the bystander activated CD8 T cell, they can produce interferon gamma. But meanwhile, also, we combined all triple cytokine. Then we found that really by the combining the triple cytokine, the activation, bystander activation is so much exaggerated, but not only this the activation, but also NK-like killing activity. Sure, only IL-15, the single treatment, it induced NK-like killing activity, but by triple combination, it's so much exaggerated. So maybe in the real human pathology, the triple cytokine, they play a role altogether in combination. So also after getting this data, I look back again, this is my HIV patient sample, and then we found that in our previous publication, we focused on IL-15, but we found that in their serum, IL-18 is also upregulated in HIV. So maybe in the real situation in HIV-infected patient, not only IL-15 alone, but also this or maybe triple or dual, this combination. Such kind of phenomenon is working in the real situation in HIV-infected patient. Yeah, so this is the end of my talk, and this in my lab. So there are the three different division. The first one is the bystander T cell activation. Second one is the COVID-19 immunology, and third one is tumor immunology. And we are working by the nationwide the clinical collaboration in South Korea. And this work was mainly done by Ho Young Lee and So Young Kim. And the final part, senescence part, was done by Se Rong Kwon in my lab. Yeah, thank you very much for your attention.